Physiology Group Project. In this video, we are going to discuss about the seed germination. At the end of this video, it gives you an idea about seed germination process and the types. So, let's move on to our video.
Seed germination is the process in which the dormant embryo of the seed resumes active growth and forms a seedling. It happens through the series of physiological and biochemical events. Now you can see the main part of the seed.
Seed germination starts with the imbibition of water by dry seed coat. Due to the imbibition, the seed coat becomes more permeable to oxygen and water. Then seed cells become enlarged. After that, it starts to respire and synthesize proteins to convert starch into sugar. Once the seed has ruptured, the radicle of the seed forms the root system and the plumule of the seed forms the shoot system. At the final stage of the germination, it grows into a seedling.
There are three types of seed germination: hypogeal seed germination, epigeal seed germination, and the viviparous seed germination. In this video, we are going to talk about those three types of seed germination. Let's talk about epigeal seed germination.
We prepared a seedling pot with soil and took good quality bean seeds. After planting the seeds, we put some water. Moisture is an essential requirement for seed germination. Firstly, we can see the radical initiation from the seeds. Epigeal germination of beans begins with radical emergence, which soon forms a branched taproot with many lateral roots. In this video, we can see cotyledons emerge from the soil surface. Now they are in soil surface level. We can see that hypocotyl elongates. Cotyledons are brought above the soil surface due to the elongation of hypocotyl. It can be seen that hypocotyls are further elongated. Also, foliage leaves form, but they are not open yet. At last, foliage leaves are open to the environment. Now they can function photosynthetically.
This is a seed. Seed contains a developing embryo, covered inside to the protected seed coat and capable of developing into another such plants. When a new plant grows from a seed, that process known as germination. During germination stage, moisture present in the soil makes the seed well and softens the seed coat. Inside the seed coat, the developing embryo lives on the fruits short in the cotyledons. First, the root appears, and later the shoot. After the leaves appear and the young plant can make absorb food, the cotyledons fall off. Tiny plant comes out of the soil where it will be able to get sunlight to obtain energy for performing photosynthesis.
Let's move to the hypogeal seed germination.
This is rice seeds. Rice seeds spray all over the soil.
so the seeds go few centimeters below the soil. First, root come out in the radicle. Seed absorb the water in 2 days. Then after, roots develop. Same time, cotyledon develop firstly, but the full leaves developed in furrow. Here we see full developed rice plant. This is first leaf and roots. Rice root is fibrous root and leaves develop in one knot.
Meaning of the hypogeel. Hypo is below and the geel is soil surface. During the process of germination, when the cotyledons remain below the soil, it is known as hypogeel germination. In this germination type, the cotyledons remains inside the soil. Seeds start to germinate, the radicle develop first. Then, shoots are developed. When shoots are developed, it come above the soil. In this germination type, the hypocotyle is small in size and hypocotyle does not elongate. It is the reason why the cotyledons remain below the soil surface and epicotyle is elongated.
The cotyledons, a large component of a seed embryo, are left in the soil. Plumule, the basic shoot tip emerging from the seed embryo, emerges above ground. The plumule pushes its way to the surface by pressing upward. Epicotyl, the seedling stem above the cotyledon, continues this process by quickly elongating its structure.
When we consider about the process of viviparous seed germination, the ovum is fertilized while still on the parent tree and grows by a combination of photosynthesis and acquisition of nutrients from the parent until it may reach a length of 50 cm. A propagule then falls to the ground. The propagules of some species root almost immediately, but others appear to have an obligatory floating period before they sink and establish themselves. The majority of floating propagules probably settle close to the parent, but long distance dispersal is also possible. Floating mangrove propagules may remain viable for a month or longer depending on current speed and direction. They could travel a considerable distance. Many mangrove species show some form of vivipary. Establishment of new mangrove plants in the unstable substrates and regular tidal washing of the mangrove environment presents a particular evolutionary challenge. All mangroves are dispersed by water and particular structures in the seed or the fruit are adapted to support flotation. In a number of groups, a degree of vivipary is observed, which is unusual in most plants other than mangrove species. The Rhizophoraceae have developed this to its fullest extent, and here the embryo grows out of the seed coat and then out of the fruit while still attached to the parent plant, so that the propagule that is eventually released is actually a seedling rather than a seed. In a number of other groups exists in which the embryo emerges from the seed coat but not the fruit prior to abscission. Longevity of seedlings is clearly important for many species.
A seed must be germinate within air and light, presence of moisture and the right temperature. Seed germination is a crucial process that influences crop yield and quality. Therefore, understanding the molecular aspects of seed dormancy and germination is the great significance for the improvement of crop yield and quality. And the seed germination determines continuous plant production necessary for human survival with regards to the supply of medicine, feed for animal grazing, and food security.